This is the last section of a very long chapter, colloids. These are things you're familiar with, but possibly not in the way we're going to describe them. Colloids are also known as dispersions, or they're called colloidal dispersions, and they're intermediate between things that are solutions, which you know as aqueous, and suspensions, all right? So let's kind of give some definitions. A solution, as you know, is homogeneous and there is absolutely no settling. Putting a size on the particles, they're anywhere from 1 to 10 angstroms. A colloid, that's what this section is about. We say that they are dispersed and they do not separate under the influence of gravity. These particles are a little bit bigger, and again, they are suspended. Well, then that makes it hard here. A suspension is the third type of material, and it is heterogeneous, and a suspension will eventually settle. So 3 years of my life were spent trying to make colloids, which tended to settle. Let's kind of put that into perspective. But the particles, there are thousands to millions of atoms, things like clay and mud, really where it happens. So examples, like I said, you do know, you have seen fog, which is a colloidal dispersion of a liquid in a gas. Think about that. You've seen smoke, which is a solid in a gas. And of course, you have drank milk, which is a solid in a liquid. So there's a whole host of different kinds of colloids. Let's focus on where it here. Look at all the different colloids. You know of all these different examples. Maybe some of you will end up in a job, maybe in marketing, maybe in research, maybe in development, maybe in customer service that has to do with something chemical, and it may be having to do with all of these materials. What crossed my mind right now is the aerosol. We never thought much about those until COVID, where the particles could be part of our exhale and cause an infection. So again, you've learned the fundamentals in this chapter. You've learned what features in simple substances give rise to different behaviors, polarity versus nonpolar. You've also learned about the colligative properties, which play a role in boiling point, freezing points, can't say osmotic pressure, but definitely in vapor pressure. So finish up this chapter. Please ask questions. Please do homework problems. No one is going to ask you to regurgitate what we've said in class. They're going to expect you to solve problems. Gen Chem aspects, okay? Colloids exhibit something called a Tyndall effect, and that is where they scatter visible light. That's what this picture is supposed to show. As you see, the light is going straight through here, but I honestly don't know what it's doing here. I think there may be a colloid. But if we call the first one a solution, there is no scattering of light. If I look at the one on the right, the milk, that is colloidal dispersion. If I were to shine a light at this, the light would push back, just like fog, your headlights when you're driving. That's really rain, all right? So here's my bad attempt at a joke. Hydrophilic versus hydrophobic. So here's the person with many, 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 many OH groups. I'd say that is a hydrophilic person. Sitting on the couch is someone who has only London groups. And the comment is, when did you first start to notice you might be hydrophobic? Hopefully you got the joke. Because hydrophilic and hydrophobic colloids. Think about oil and water. Think about Italian salad dressing. You have olive oil and you have water. You may have some vinegar. Oil, which is nonpolar, naturally separates from water. Why does this happen? Well, you've had chapter 11, intermolecular forces. You've had this chapter, polar versus nonpolar materials. If I want to suspend oil and water, I will use an additive. Sometimes they're called surfactants. Sometimes they're called detergents. There's many different names. So colloids can be hydrophilic, which means loving. And that could be something like an enzyme or an antibody in our bloodstream. Or they could be hydrophobic, which is water hating. So as you look at these pictures, here it says hydrophobic particle. I'm going to put the word L on there, or let's say L and say London forces. I'm going to say very nonpolar. What about the blue? Well, I'm going to say hydrogen bonding and I'm going to say polar. So as you see, there's a demarcation between the two, and what happens is if we put something on the surface, what can happen is we can suspend the particle. Let's say Miss Frizzle Goes to the Waterworks. I don't know if you've read that book, but if you've read that book, you know exactly what I'm talking about. So instead of that cartoon version, what about this real version? Here I have an N, which is going to impart some polarity. Here I have an O, which is going to impart polarity. And we have a couple of these sitting on a non-polar substance. So this could either be the additive or it could be the material that you're trying to take out of solution. It's a very vast field. If I want to stabilize a hydrophilic colloid in water, that is something that is non-polar, I can use a molecule, I can use an additive like sodium stearate, and there is the formula for sodium stearate. I'm just going to draw a line because there is a tail that is non-polar, and that will solubilize the oil. There is a head that is polar that will interact with the water. And that's what this picture is showing. This is a non-polar thing like oil. What we have here is the non-polar tail of the additive, and what we have over here is the head, which is very, very polar. Now you'll say, why do you even bother to tell us in class? Think about detergents, think about shampoo, think about all the different foods where they make an emulsion. This is real life chemistry, and to me this is why I became a scientist, not for all the stuff that came before this, but because I'm curious, how do things work? In real life, you can take this either further. Proteins, they can be folded or they can be unfolded by denaturization. They require a specific conformation in order to do what they're supposed to do, say as an enzyme. So you will hear about this in your other classes, and essentially what I want to drive home is if you have something, it can have a hydrophobic portion, which is nonpolar, and it can have a hydrophilic portion, which is polar. What do I have left? Well, sickle cell anemia is a nice example as we talk about this. In normal hemoglobin, you have this straight chain. In abnormal, what you have here is you have a methyl group. And that methyl group is not a straight chain. And what it does is it causes the red blood cells to change their shape. Okay, so you'll probably learn about this if you're going into a medical field. On the last page, I have a picture of my own research. When I worked at British Petroleum, I worked on aluminum nitride. And as you can see, we didn't have fancy things, and you can see how old I am. We had to just draw them and type them. But this was desirable. What we wanted was the aluminum nitride to be dispersed in our it was called a slurry. So again, there's so many more names. But what was not desirable is what I would cry about all the time, because I would come into lab, I would set up a number of experiments, and what would happen is these particles would do something called flocculate. Flocculate is where they would come together and become bigger, and because they became bigger, they fell due to gravity, and it was not very pretty at all. So that's, again, Miss Frizzle Goes to the Waterworks. Colloids are really important in the real world. We talked about milk. I'm going to go